In a Berkeley laboratory, Rebecca Cann was extracting DNA from an object most people would never imagine becoming a map of humanity: the human placenta. The samples came from women with roots across Africa, Asia, Europe, Australia, and New Guinea. When Cann and her colleagues compared them, every maternal branch eventually converged on one African woman. Not one race, not one nation. One maternal ancestor. That was the discovery behind the idea that only one DNA line matched everyone on Earth. But scientists did not sequence every person alive or match complete human genomes. They followed one unusually durable piece of DNA passed from mothers to their children. The real mystery was how one woman's line could reach every living person, while countless other lines vanished. Its answer would turn a story about one miraculous mother into something much larger about how ancestry survives. Most of your DNA sits inside the nucleus of each cell, mixed and reshuffled from both parents. Mitochondria carry a much smaller genetic record of their own. In ordinary human inheritance, that record comes through the mother. A son receives it, but he does not normally pass it forward. A daughter can. That simple pathway gave Cann's team a clean trail. Small changes accumulate along maternal lines. People who share a recent maternal ancestor carry more similar patterns. People whose lines separated earlier carry more differences. Arrange those differences as branches, and DNA becomes a family tree. In 1987, the team's landmark paper compared mitochondrial DNA from 147 people belonging to 5 broad geographic populations. The sample was global for its time, but it was not every human being, and it covered only this narrow maternal record. Even so, its shape was startling. The tree did not split humanity into separate continental origins. Its deepest branching point lay in Africa, and every sample ultimately joined the same maternal trunk. The map had found one root. Then a nickname turned that root into a myth. Mitochondrial Eve made the result sound far stranger than it was. Picture 2 women living in the same ancient community. Both have children. Both leave descendants. But one has daughters, while the other's line continues only through sons. The families survive. Only one mitochondrial line continues. That is the severe logic of maternal inheritance. A son receives his mother's mitochondrial DNA, but he does not normally pass it forward. If a woman's daughters have no daughters, her marker disappears 1 generation later. No massacre is required. No population has to vanish. Across immense spans of time, ordinary births, deaths, movement, and chance prune the tree. Geneticists call part of that process drift. This is why an extinct mitochondrial branch does not mean an extinct family. The woman who carried it may still have living descendants through sons, grandsons, and countless other routes. Her ancestry survives inside people even after this one genetic signature is gone. Follow the maternal branches that did survive backward, and eventually they meet. The most recent woman at that junction is mitochondrial Eve. Current estimates place her around 150,000 years ago in Africa, but she was not the first modern human, the only woman alive, or the source of most of anyone's genome. She lived inside a much larger population. Her distinction came later. At least 2 of her maternal branches endured, while equivalent branches from her contemporaries ended. Eve was not humanity's only mother. She was the woman whose maternal road never broke. And the location of that junction supplied the next clue. A family tree carries time inside its shape. Deep splits mark lineages that separated long ago. Africa contains the richest and deepest diversity of human mitochondrial lineages. The major lines outside Africa do not stand beside them as an equally old tree. They grow from within a younger part of the African one. That pattern turns geography into history. When some human groups moved beyond Africa, they carried only part of the continent's older maternal diversity with them. Their descendants spread across coasts, inland corridors, mountains, and eventually oceans, producing new branches along the way. The map widened. The route did not move. This placed the maternal lineages now found across Europe, Asia, Oceania, and the Americas inside a deeper African story. But it did not point to one modern nation or one tiny birthplace. Later evidence showed that early Homo sapiens lived in structured populations across Africa, sometimes separated and sometimes reconnecting, long before a subset expanded outward. The deepest branches therefore preserve movement within Africa before they record movement beyond it. They do not crown a modern people as the original humans. They show that the global journey began inside a continent already filled with human history. That was a major answer. Then another kind of DNA exposed real ancestors missing from this elegant tree. Inside Neanderthal bones, nuclear DNA revealed an apparent contradiction. Many living people with ancestry outside Africa carry fragments inherited from Neanderthal encounters. Their ancestry survived. Yet no surviving Neanderthal mitochondrial lineage has been identified in living humans. The contradiction disappears once one family is imagined. A Neanderthal woman could have a son whose descendants continued for thousands of generations. Her nuclear DNA might pass onward in smaller and smaller fragments. Her mitochondrial DNA would stop at that son because he could not normally transmit it. Another maternal branch could continue longer, then end when daughters stopped having daughters. A real ancestor remains in the family and disappears from the maternal map. Scientists cannot name one universal reason every Neanderthal mitochondrial line was lost. They do not need one. The contrast shows the larger point. Absence from the mitochondrial tree is not absence from human ancestry. This is what the simple image of Eve hides. Mitochondrial DNA keeps one root astonishingly clear by ignoring almost everything else. Your sequence followed your mother, her mother, and the mothers before them. But your wider family tree includes fathers, grandfathers, cousins, migrants, and ancient encounters that this marker cannot record. The maternal root was real. It was never a complete portrait. And when scientists traced a paternal marker, the imagined founding couple disappeared too. The Y chromosome preserves a different straight path, from fathers to sons. Follow its surviving branches backward, and they converge on a common ancestor nicknamed Y chromosomal Adam. The names built a couple that genetics never found. There is no reason to think Adam and Eve were partners, neighbors, or even exact contemporaries. Their estimated time ranges may overlap, but they are simply the roots of two separate inheritance systems. Change the genetic marker, and the ancestral tree changes with it. Most of the nuclear genome refuses to form any single clean tree. It is reshuffled every generation, breaking ancestry into segments with different histories. One stretch can lead toward an ancient African population, another toward a later migration, and another toward an encounter with an archaic human group. Every piece has its own route backward. Mitochondrial Eve occupies only one of those routes. Her line appears universal because maternal inheritance kept that trail unusually straight, while countless neighboring trails crossed, split, and vanished from view. The idea of one ancestral couple was gone. One harder test remained, whether a much larger map would preserve even her route. The weakest part of the first global map, was the very continent at its root. Africa held the deepest maternal diversity, yet many communities were barely represented. Any missing lineage could add a branch, alter a date, or move an early split. So the map faced a far harder test. By 2026, a new analysis drew on nearly 5,000 complete mitochondrial genomes from Africa, including many previously underrepresented parts of sub-Saharan Africa. The added sequences redrew branches and exposed movement Cann's first tree could not see. They sharpened Africa's internal history, but they did not produce a second root. The deepest division still lay inside Africa. Dates can shift as rare lineages appear and genetic clocks improve. The central structure held. Better data clarified the limit. It did not reconstruct Eve's complete genome or assign her to a modern population. The root is a junction inferred from living maternal lines, not a preserved woman waiting to be matched. The first map was incomplete. Its central discovery survived. And that made the meaning of the only match almost the opposite of the myth built around it. So what was the only match? It was not one modern person whose genome matched everyone. It was not one tribe, carrying the original human DNA. And it was never proof that humanity began with a solitary woman. The match was one surviving maternal thread, the single point at which the mitochondrial line of every living person can be joined. Rebecca Cann found its outline in DNA taken from placentas. Larger maps have added thousands of branches, but they still lead into the same deep African tree. That is what makes the result powerful. One maternal root does not make humanity a single bloodline. It places every living population on the same inherited thread, while the rest of human ancestry remains a web. Mitochondrial Eve matched the world only because so many other maternal lines stopped. Behind that single line remain countless women and men, migrations and encounters, preserved in other parts of our ancestry. The one match is real. But humanity was never one line alone. Which discovery should we trace next? Y chromosomal Adam, the lost Neanderthal maternal lines, or the newest map of Africa's DNA? Share your choice below and subscribe for the next story hidden inside human ancestry.